Good morning and welcome to Chapter 3. In Chapter 3, we're going to be covering cell structure and function. And we're going to start this by defining what a prokaryote is. Okay. So prokaryotes are typically bacteria and archaea. And we are not going to be discussing archaea in this course because they do not have clinical significance. So we're going to be emphasizing our attention on bacteria. So what's the main distinction between prokaryotes and eukaryotes? Well, as the name implies, a prokaryote is so early on in evolution that it is absent a nucleus. So prokarya means before the nucleus. Eukaryote, on the other hand, means that is late on in evolution and it has a true nucleus. Eukaryote. The second distinction between prokaryotes and eukaryotes is the complexity of the organism. So prokaryotes are very simple. They do not have complex organelles. Eukaryotes do. So eukaryotes have membrane-bound organelles such as the rough and the smooth endoplasmic reticulum, the Golgi complex, mitochondria, chloroplasts, etc. Prokaryotes do not have these complex membrane-bound organelles. Another distinction is the size. Prokaryotes are much smaller than eukaryotes. Your author says that prokaryotes are typically less than 1 micrometer. By contrast, a eukaryote can be 1,000 or 10,000 times bigger than a prokaryote. Okay, and then the last major distinction is the level of organization. So prokaryotes typically only organize themselves in colonies. In contrast, eukaryotes can organize themselves in colonies. They can organize themselves in tissues, in organs and organelles, and in organisms, which is what makes us be present in the world. So we are eukaryotic animals. So those are the four main distinctions, okay? Presence and absence of a nucleus, presence and absence of membrane-bound organelles, the size, and the complexity of organization. Okay. Now, as we move out to the external structures of prokaryotes, the first thing that we start with is the glycocalyx. Okay. So glyco means sugar. Calyx essentially is a covering. So this is the sugary covering. Okay. If you've ever accidentally spilled honey or something that is rich in sugar somewhere, you find out very quickly that it is very, very sticky. Glycocalyces are very, very sticky. And so prokaryotes use these glycocalyces as a means to attach to surfaces or as means to attach to each other, okay? And when they attach to each other, they form these complex structures called biofilms. And biofilms is a significant problem in the clinical environment because once they form, they're very hard to break up and they're very hard to treat. Okay, so glycocalysis is a precursor to these biofilms. Okay, there's two major types of glycocalysis. The capsule, okay. What's the significance of the capsule? Is the fact that it provides protection to the bacteria that has the capsule. Okay. The other main type of glycocalyx is the slime layer. And the significance of the slime layer is, again, it has that sugary coating that causes it to be very, very sticky and allows it to attach to the environment, structures, walls, catheters, and of course to each other. Okay. So here's some examples. Okay, this is Streptococcus. You can see what a thick glycocalyx it has. This is a capsule, and consequently, it is very, very hard to treat this, because if you're giving it, if you're taking antibiotics, it's difficult for those antibiotics to diffuse through that thick glycocalyx capsule. There's a slime layer. Okay, as you can see, the slime layer is not as structured, and again, it is repeating sugar units, but it allows this organism to attach to its environment and to each other very well. Another external structure is flagella. Flagella is what allows cells to be motile, or gives them the ability to move. And the reason that flagella are important to us is because they help us characterize or identify the microorganism. Okay, different bacteria have different types of flagella, and they have different organizations of flagella. So if you can see the type and organization of flagella, that can help you identify the organism. Okay, one of the things that's important for you to know in this chapter is the parts of the flagella. Okay, the flagella consists of the filament, the hook, and the basal body. What does that mean? Well, here's the filament. The filament is literally the part that turns. Okay, the hook, as the name implies, is crooked, and it attaches the filament to the basal body, where the basal body is essentially the engine. Okay, and it is in fact an engine. It's a engine that's driven via a proton gradient. Okay, so another thing that's interesting to note, okay, is that the structure of this basal body is different in the different types of gram bacteria. So here's a gram-positive microorganism. Here's a gram-negative microorganism. We're going to talk about this again, because this is an important point, okay? So in the gram-positive, you have this thick peptidoglycan layer and a thin cytoplasmic membrane inside. In the gram-negative microorganism, you have a thin peptidoglycan cell wall in the middle, and you have an outer and inner membrane. So you have an outer membrane and a cytoplasmic membrane. Why does this matter? It matters because the proton gradient that's necessary to drive this basal body, this engine, can only be accumulated in an environment that has a plasma membrane. So notice that in the gram-positive organism, there's two protein rings, okay? If you can imagine, these are the turbines that are being turned, and they can only be where the cytoplasmic membrane is at. In contrast, in the gram-negative microorganism, because it has not one but two membranes, there's not one but two sets of rings. One with a cytoplasmic membrane inside, and one with the outer membrane on the outside. And the artist, okay, they made a mistake. This ring is not supposed to be in the middle, and the peptidoglycan layer should be up here with the other ring. So this is an instance where the drawing is not accurate, okay? But again, the structure of the flagella helps you discern whether you are dealing with what's called a gram-positive organism or a gram-negative organism. Okay, now we're going to go into the organization of the flagella, okay? So notice that this organism right here has flagella all the way around it. We call this a peritrichous flagella, peri meaning surrounding. So the entire organism is surrounded by flagella. And by contrast, this one in the middle has a single flagella, and that's called monotrichous, mono meaning one. So one flagella, monotrichous. And then this one has a little tuft of flagella, okay? So this one is called lophotrichous, meaning that we have low tuft. What does a tuft mean? It means more than one. So you can see that there's 1, 2, 3, maybe 4 flagella there. So this is a Lophotrichus bacterium. Okay. Go to the next slide. Here's additional examples. Here's our Monotrichus. Here's our Lophotrichus. Here's our Peritrichus. And then the author introduces yet a fourth one. This is called Amphitrichus. Amphi means both. Why? Because the flagella comes out of both ends of the microorganism, Amphitrichus. And Helicobacter pylori and syphilis are like this. They have an Amphitrichus flagella that spirals around the organism, and the way that the organism travels is by spiraling through the environment. Okay, here's a better example of this. You can see even better. Okay, so here it's called an axial filament because it comes out the ends, but it also goes all the way down the axis of the microorganism. Again, examples of these is syphilis and Helicobacter pylori. And these guys can twirl and consequently burrow themselves into the mucal lining of epithelium or the mucal lining of the stomach in the case of Helicobacter pylori. So this is this is an instance where the locomotion of the microorganism makes it just a little bit scarier, a little bit more dangerous. Moving along, we have these things called fimbriae, okay? So fimbriae are long bristle-like projections. Here's an example of fimbriae. See how incredibly long they are? Okay. This allows the organism to attach to its environment even better. Okay. So before we said that the glycocalyx is an important precursor to biofilms, now I'm telling you that fimbriae are also an important precursor to the production of these biofilms. What's a biofilm? Remember, a biofilm is when two or more microorganisms stick to each other, okay, and consequently start functioning against their host synergistically. And that gets a little bit scary because then it's harder to treat. I'm not going to go into pili. Pili is the way that bacteria carry out conjugation. Okay, they transfer genetic material, which are called plasmids, through pili. We're going to go into that later on in the course. For now, what I'm interested in you remembering is the glycocalyx and the fimbriae, again, because they're important precursors to the formation of this biofilm. Okay, and if you look at Neisseria gonorrhoeae, the causative agent of gonorrhea, you would see very well-defined fimbriae in it. And the other thing that's interesting about the fimbriae is that it's not arbitrary what they stick to. They stick to very specific receptors. So Neisseria gonorrhoeae, for example, will not stick to just any epithelium. It can only stick to epithelium that has receptors that are recognized by these fimbriae. And that's why infections of gonorrhea only happen in the urogenital opening, in the rectum, and in the pharynx. It doesn't happen anywhere in the body. It's only in those three places. And it's this fimbriae that characterizes where these organisms choose to stick to and cause an infection. Okay. Let's go past pili here. Okay, bacterial cell walls. So the bacterial cell wall, again, I addressed a little bit about what gram-positive, gram-negative look like. Okay, here's the components of gram-positive peptidoglycan. Okay, is this N-acetylglucosamine and this N-acetylmuramic acid. Call it NAG and NAM. So you have alternating repeating units of these two modified sugars, and that repeating structure makes the building block of this so-called peptidoglycan cell wall. So this pink structure right here, that's peptidoglycan. Okay, and the fact that it's very thick here tells us that this is a gram-positive organism. The fact that it's very thin over here tells us that it's a gram-negative organism. So what else do we have? You have mycolic acids in addition to peptidoglycan cell wall. The mycolic acid is hydrophobic, and it doesn't allow the bacteria to be traversed by anything that is water soluble. And that's important because it protects the bacteria even more, okay? So this is important. When an organism has 60% or more of this hydrophobic mycolic acid, it is said to be acid fast. And again, it protects it against desiccation, and it protects it against anything that would dissolve in water. We also have these teichoic acids. The teichoic acids, as you can see here, are waxy acids that tether the thick peptidoglycan cell wall to the cytoplasmic membrane of the gram-positive organism. So it's important for you to know that the cell walls can be peptidoglycan in gram-positive and gram-negative organisms, that they are mycolic acids in acid-fast microorganisms, and finally that teichoic acids tether both the peptidoglycan cell wall and the mycolic acids onto the inner membrane in these gram-positive and acid-fast microorganisms. Here's a gram-negative microorganism. Notice again that we have a very thin peptidoglycan cell wall, and then we have an inner cytoplasmic membrane and an external cytoplasmic membrane. So gram-negative have essentially three components to their cell wall: two plasma membranes and a thin peptidoglycan cell wall. Okay, let me see. We're going to say that that's the end of this lecture. Okay, I hope you learned a little bit. I know that I'm going fast, but I'm trying to keep these videos short so that they don't bore you. Have a wonderful day, and until next time, have fun.